Everyone, and welcome to our video on the movement of water through plants. This is the third video in our Transporting Plants series, and what we're going to be doing in this one is having a look at our Spec Reference 3.1.3d, which is the transport of water into the plant, through the plant and to the air surrounding the leaves, which includes the details of those key pathways the water will take. So let's jump right in then to the names of those three pathways that we are going to need to know about. When we're considering how water moves through a plant, it will either travel through the Apoplast Pathway, the Symplast Pathway, or the Vacuolar Pathway. And what we'll do is we'll take each one of those in turn to see what we actually mean by it and how the water is going to travel. So our first pathway, the Apoplast Pathway. What we're looking at here is that the water is actually going to pass between the cell walls and between the cells through this process of capillary action. Now, what we mean by this is that it's not actually going to go through any plasma membranes fully into those cells. So if we have a little look at the diagram in the top corner here, we can see it with the little green arrows on this one. So what it's actually doing is it's just moving between those cell walls, between each of those cells there, but it's not actually passing through the plasma membrane into the cytoplasm. So when we're looking at how the water is moving here, it is through that process of mass flow, which, if you remember from our earlier work, that is just how we refer to the bulk movement of those materials. So Apoplast pathway is basically between the cell walls by that capillary action. Key thing about this is that it is faster than our other main pathway, the Symplast pathway. Now, this isn't something that can carry on indefinitely in our plant. Eventually, we get to this little region called the Casparian strip, which is basically a waxy band of this material called suberin, which we find in the cell wall. And this is going to occur at the endodermis. Now, what that means is we're able to go through the Apoplast pathway until we get to the endodermis, hit that Casparian strip, and at that point, the water is forced into the Symplast pathway. We can't keep moving through Apoplast anymore. So pathway option number 2 is the Symplast pathway itself. And in the Symplast pathway, which are the red arrows on the little diagram in the top right there, this is where the water enters the cell cytoplasm. So in order to do that, it obviously has to cross the plasma membrane. Now, what we then find is once that water has actually entered the cell cytoplasm through that plasma membrane in that first cell, it's able to pass through those structures called the plasmodesmata, which goes from one cell to the next. So what we're finding here is that our water is going to move down that water potential gradient by the process of osmosis. Key thing to remember is Symplast is slower than the Apoplast pathway. Third and final one, this one doesn't come up as often as the others. You might get a 1 mark throwaway in another question that, to be honest, you can get the marking points talking about Apoplast or Symplast. But our third pathway, for the sake of completion, is our Vacuolar pathway. Now, the name kind of gives away what it does here, is that the water is going to pass through the vacuoles in this case. So you can see it's represented by the blue arrows on our little diagram at the top. What we do need to understand before we leave this whole idea of water moving through our plants is a little bit more about our water potential, because this is something we've mentioned at a few points across not only your A-level course but back at GCSE as well. But we need to make sure we really understand what we're talking about when it comes to water potential. So first of all, when we talk about water potential, it's a measure of the tendency of our water molecules to move from one place to another. And hopefully what we do remember as our little definition of how water is going to move, it's going to go from that area of higher water potential, which we can describe as being less negative, to an area of lower water potential or a more negative region. So the water is moving down that water potential gradient. Now we can use our little abbreviation there, which looks kind of like a little trident. It's a Greek letter, of course, and that is our water potential. Now, if we consider pure water, that has got a water potential of 0. Okay, so when we're talking pure water, that is a water potential of 0. So that would be obviously a very high water potential because it is a non-negative number. It's less negative than any other number. If we now dissolve some solutes in that water, what we're going to see is the water potential is going to become more negative. So basically it moves further into the negative region, further away from the 0. So pure water is 0. The more solutes we dissolve, the more negative that water potential becomes. If we consider what this means in terms of movement of water as far as our plant cells go, if we place plant cells in pure water, then water is going to enter those plant cells. We can all think back to that lovely potato experiment you probably did at GCSE, little bits of potato in different concentrations. The ones you put in pure water were the ones that gained in mass or gained in length, whatever it may have been that you were measuring. Whole reason, water has entered those plant cells. The reason that they're going to do this change is that as that water enters the cells, they are going to exert pressure on the cell wall. So what we find here is that as that pressure potential builds up, then we're going to reduce that movement in of the water. Eventually we get to a point where it's going to actually be classified as being full of water. So that cell is not going to be able to basically expand any further because of the rigid nature of our cell wall. And at that point, we describe our cell as being turgid. If we now do the opposite, we place our plant cells in a solution with a very negative water potential. So this would be the example of your quite concentrated sugar or salt solution, for example. That would have a very negative water potential. What we find is water is going to leave the plant cells and move into the solution. So they lose water. As that water is leaving the cells, what we find is our cytoplasm and the vacuoles are going to start to shrink, and that means our cytoplasm is eventually going to stop pushing out against the cell wall. At that point, our cell is no longer turgid because we've lost that pressure against our cell wall. If water continues to move out beyond that point, that plasma membrane is actually going to pull away from the cell wall in this process called plasmolysis. At this point, we describe our tissue as being flaccid. Just to put that into context in terms of seeing some numbers so we can understand the direction of the movement then. What we've got here are two different cells or regions. The region on the left here has a water potential of minus 800 kilopascals, and on the right, minus 1,200 kilopascals. So remember, water is going to go from the less negative water potential to the more negative water potential through that process of osmosis. So because minus 800 is closer to 0 than minus 1,200, then water is going to be flowing from the left, the minus 800, to the right, minus 1,200. So just make sure that you are thinking in terms of the degree of negativity. So the more negative it is, then that is going to lead to water moving in that direction, towards the more negative region. As always, I do suggest that you subscribe to the channel so you can see when another video is uploaded, and of course head on over to the A-Level Biology website where you can see a range of other resources designed to help you with your A-Level studies.